Continuiamo il
We
nostro
now
viaggio.
continue our journey, this time with a look inside P.R.M. pharmacology. Physiological regulating medicine is founded on three major pillars. The first pillar is represented by the homeopathic tradition and by homotoxicology. The second pillar, which we have already seen, is identified with psychoneuroendocrino-immunology. We have still not encountered the third pillar, but it is just as important as the others, and it deals with the remedies that constitute nutritional supplements. Why is this pillar also so important? Well, the hormones, neuropeptides and interleukins, for example, are protein structures, and is therefore necessary to provide the body with the bricks with which these substances are constructed, the amino acids. Does this mean that vitamins and trace elements are not important to increasing enzymatic function? In a final analysis, our therapy, which is a biological therapy, travels along the track of enzymatic stimulation. So, the coenzymatic activity of vitamins or trace elements becomes fundamental. We love to talk about the tool bag in pharmacology. Well, the tool bag for physiological regulating medicine is really quite rich. Sometimes we draw from this bag by fishing for typical homeopathic remedies such as plant, mineral or animal substances. And sometimes we draw freely from the homotoxicology sub-bag of tools. For example, we use pig organ derivatives or intermediate catalysts or even NOSODES. We have already understood how important it is to draw from the tool bag of physiological regulating medicine, and in particular PNEI, hormones, cytokines, etc. What is extraordinary is that the players of P.R.M. pharmacology, i.e. the pharmaceuticals, are none other than a perfect assembly of these individual components. We often love to speak of the dream team, and every drug is a team, wherein a plant homeopathic remedy finds its maximum expression because an intermediate catalyst of the KREBS cycle is present, and of course, since an interleukin that enhances its action is present. One example that I believe is very explanatory in this sense is one of the most well-known pharmaceuticals, called GUNA FLAM. So, let's get to know this drug, our broad-spectrum anti-inflammatory. You can note in this structure that we like to refer to as the pharmaceutical explosion that various groups of components are present. One of these groups, as you can see, is represented by typical remedies of classic homeopathy. For example, aconitum and belladonna. Alongside these remedies, whose functional mechanism we will see, the typical remedies of PNEI are also present. For example, anti-interleukin-1 or TGF-beta, or even interleukin-10 with its anti-inflammatory action, and there are also trace elements present, such as copper. Here I believe that it is very easy to observe how, in an individual drug as in a dream team, all of the components play together to beat the enemy, and in this case, our enemy is disease. Behind every P.R.M. pharmaceutical, there is a profound homeopharmacologic study. Each drug is designed in the fundamental light of two principles, and the first principle is described in three terms: synergism, balance, and completeness of action. What does each of these terms mean? Synergism means that the remedies contained in each of the P.R.M. pharmaceuticals altogether, like a team, lead to a superior result with respect to the action of every single component. What does balance mean? That every component is present in a precise dilution, or is present for a particular action in relation to other components, so that the action of the pharmaceutical is generally mild. For example, in Cytomix, a drug with an immunostimulatory action, many interleukins with Th1 polarity, and therefore stimulatory effects to the immune response, are present. But there is also an interleukin, interleukin-4, with Th2 polarity present. Why was it included? So that an overly strong stimulation of Th1 could thereby be avoided. The third term is completeness of action. The included remedies are chosen in order to cover all of the different extrinsic symptoms that accompany a certain disease. The second principle that has guided the composition and formulation of pharmaceuticals for physiological regulating medicine is even more interesting. Its statement is this: the etiopathogenetic path of the disease that a P.R.M. pharmaceutical is meant to cure is inscribed in its pharmacological structure. To say it in a much simpler way, this etiopathogenetic path is reconstructed step by step with specific remedies, beginning with the study of the etiopathogenesis of a disease. To explain it very concisely with a simple example, the causes of gastritis with acidic hypersecretion can be different, ranging from alcohol to food. Alcohol or food, i.e. the stressor of the gastric mucosa, determines an alteration in acid secretion by the parietal cell. Too much hydrochloric acid, which inflames the gastric mucosa with accompanying typical gastritis symptomatology, is produced. At the same time, the mucosa responds with a defensive catarrh reaction, which is appropriate but up to a certain point, and if not treated properly, this gastritis can progress along its pathogenic path towards chronic gastritis or toward a peptic ulcer. This is a stereotypical path of gastritis with acid hypersecretion. We will note that the GUNA stomach pharmaceutical, which is specific for the therapy of gastritis symptoms, contains remedies that saturate each of the etiopathogenetic steps of acid secretion gastritis. We have remedies that regulate acid secretion, remedies that symptomatically reduce mucosal inflammation, and remedies that modulate the catarrh reaction, which, if it reaches a certain extent, could become the ideal sustenance for the proliferation, for example, of Helicobacter pylori. A complete pharmaceutical would also, therefore, be capable of draining excess mucus. Finally, we have some remedies such as epidermal growth factor that are capable of inducing a proliferation of the mucosal epithelial cells and therefore counteracting the pathogenic evolution that could lead perhaps to a peptic ulcer. What have we drawn from classic homeopathy? Well, classic homeopathic pharmacology involves the use of plant, mineral, or animal remedies in accordance with the two fundamental principles of homeopathy. The first is the principle of infinitesimal doses, or the inversion of the effect, and the second is the principle of similarity. Very briefly, let's try to understand what these two principles mean. I'd like to present this very interesting experiment that shows how the first principle of homeopathy, the principle of infinitesimal doses, is absolutely true. What does this principle mean? That a substance inverts its effect according to the doses with which it's used. In fact, not all substances are subject to this principle, but those that are used according to the homeopathic pharmacopoeia are, and this is a mechanism of action that these substances carry out within that extraordinary team that represents a complex preparation of P.R.M. In this their experimental work, you can observe the effects of different concentrations of an alkaloid, aconitine, on the isolated and perfused heart of an eel. At a molar concentration of 10 to the power of minus 5, aconitine induces a biological response of tachycardia, or fibrillation in the eel heart. When the same substance, aconitine, is no longer at a molar concentration of 10 to the power of minus 5, but is instead more diluted at 10 to the power of minus 7, its effects are inverted, and it no longer induces fibrillation, but its opposite, bradycardia. One can see that it is even more extraordinary to observe the effects of a 10 to the power of minus 18 molar concentration of the same substance, the same alkaloid, aconitine, on the isolated and perfused eel heart. This substance, at a molar concentration of 10 to the power of minus 18, is able to restore the proper cardiac rhythm in an ill heart that was previously pre-intoxicated by massive doses of aconitine at a molar concentration of 10 to the power of -5. So, what the father of homeopathy, Hahnemann, had intuited is really true. It is not only the quality of the substance that is important, but also the quantity that we use. That is, the same substance is able to induce a pathological profile in a healthy subject if used in large quantities, and is able to cure that very same symptomatic profile in a sick subject if used in extremely low doses. To make a very simple, if somewhat forced example, we can talk about the effects of water and the effects of a poison such as strychnine. If we use water in very large quantities, we could, in a healthy subject, cause an alteration of his hydroelectrolytic equilibrium, leading to the pathological condition of alkalosis and therefore guiding him to his death. Yet, water is the most inert substance, the least evil that we know. Conversely, a poison such as strychnine, the famous poison of Agatha Christie's mysteries, or arsenic, can heal if used at very low doses. This is exactly what we do with strychnine. In homeopathy, strychnine is used to cure acid hypersecretion gastritis. How are we able to say such a thing? Because if we use high doses of strychnine, extracts from a plant called Nux vomica, we induce a case of acid hypersecretion gastritis. In what way? We would hyperstimulate the acetylcholine receptor on the parietal cells of the gastric mucosal glands. The proton pump would work in an excessive way. Too much hydrochloric acid will be produced, and this hydrochloric acid will be responsible for those symptoms that we all know as heartburn and gastric acidity. However, in accordance with the principle of inversion of effect, it should occur that with extremely low doses of strychnine, that is of Nux vomica, or if we prefer, we could say homeopathic dosages of Strynos Nux vomica, its effect is reversed, and this is what is observed experimentally. Instead of stimulating the acetylcholine receptor on the parietal cell, low homeopathic doses of strychnine halt the receptor activity. The result is biological regulation of the proton pump, and therefore biological regulation of acid secretion. In short, not only the quality of the substance is important, but in homeopathy, the quantity of the substance used becomes fundamental, because certain substances, not all but many, are able to invert their effect. This is the magic that is not actually magic, but the science of homeopathy. After having visited the therapeutic arsenal of classic homeopathy, our journey continues as we make a leap into the homotoxicology tool bag. We refer to 3 classes of remedies in physiological regulating medicine by looking at the homotoxicology tradition. Intermediate catalysts, the metabolic stimulators, NOSODES, the immunological stimulators, and pig organ derivatives, the stimulators of organ function. Let's see very briefly how each of these classes of remedies functions. First, we have said that the intermediate catalysts are metabolic stimulators and are able to restart a slowed or sometimes even a blocked KREBS cycle with their enzymatic induction mechanism. As you will recall, the KREBS cycle represents the nuclear power plant of our cell within the mitochondrion, and it is in the mitochondrion that the fuel of our organism, ATP, is produced. The production of ATP is a refining process that starts with the elementary macromolecules, carbohydrates, lipids, and proteins, and must break them up into smaller and smaller pieces until producing 4 pairs of hydrogen atoms, which then enter the electron transport chain, extending until the oxygen molecule that they will reduce to water. And it is in the various passages that the green fuel, that is ATP, is formed. In some diseases, especially in functional disorders and in chronic degenerative diseases, blocks may exist in the KREBS cycle that are tied to cellular intoxication or a deficiency of some of the Krebs cycle intermediates. Using the substrates, the salts of the Krebs cycle, in a homeopathic dilution stimulates the function of an enzyme that is, let's say, lazy in that moment. In this case, we don't refer to homeopathy, but to biochemistry. In fact, the Krebs cycle intermediates that we use in our pharmaceuticals at a homeopathic concentration, in accordance with the law of enzymatic kinetics proposed by Michaelis-Menten, can function as inductors for their specific enzyme. Remember, the Michaelis-Menten law says that in a reaction catalyzed by an enzyme, the reaction velocity is inversely proportional to the concentration of the enzymatic substrate. Wow! We use enzymatic substrates in a homeopathic dilution that is extremely diluted. So, this low molecular concentration functions as substrate, and we are able to restart a Krebs cycle that could have been said to be lazy or slow. We now know, after years of study, what the block in the Krebs cycle is and where it is located for every pathology, or at least for a large number of diseases. This is the reason for which we find some catalysts in certain P.R.M. drugs, and in other drugs other catalysts, each specific to the treatment of a specific functional disease, especially for chronic degenerative diseases. Next to the intermediate catalysts, a broad page in the P.R.M. pharmacology is written by those players in homotoxicology that are called NOSODES. NOSODES are derivatives from pathological tissues, as well as from excretions, secretions, and microbial cultures. In short, we can say that everything that is antigenic can become a nosode. As an antigen, a nosode is capable of stimulating the immune system, and in particular, we now know that a nosode is able to stimulate the reticuloendothelial system, or you may prefer to call it the macrophage histiocytic system. That is, the part of the immune system that is not circulating, but is allocated to a tissue called the extracellular matrix, which we will discuss at length in this course. I would like to highlight a very important aspect. When we use a nosode, and we are therefore stimulating the immune system, we do not produce other antibodies, but these are the immunocompetent cells that are stimulated by nosodic action. T helper lymphocytes, macrophages, cytotoxic T lymphocytes, and natural killer cells. Only a very small percentage of antibodies are stimulated by the nosodes. This is the big difference between a vaccine and a nosode. A vaccine induces a proliferation of antibodies, and a nosode produces a cell-mediated response. Let's never forget this fundamental aspect. For example, we know another P.R.M. pharmaceutical called GUNA flu, which is a well-known pharmaceutical and is used for the prevention and treatment of colds and the flu. As you will be able to see, its complex structure is based on an immune stimulation core. There are 3 substances, and 2 of these substances are nosodes. One is called Annus barbariae, and the other is called influenzinum. These are the 2 nosodes, and they are obtained from the influenza viral strain, the viral strain being an orthomyxovirus. Why is GUNA flu, with the 2 nosodes it contains, so effective? Well, we can discuss it in a minute, and in the meantime we can get to know the drug. As you know, the influenza virus is an orthomyxovirus, and you know that an orthomyxovirus has a particular feature, its genetic instability. What does this mean? It means that this virus is able to drift and shift, and in very simple words, is able to disguise itself. When we use an anti-influenza vaccine, we stimulate the proliferation of antibodies. The antibodies have 2 characteristics. Memory. I was vaccinated for polio when I was a child, and I'm still covered. And the other characteristic is specificity. This is very important. Every antibody is specific for a particular antigen. However, If the virus, that is the antigen, is able to drift or shift, i.e. to disguise itself, you can understand that the antibody stimulated by the vaccine is not able to neutralize it. This is a reason for which some anti-influenza vaccines have an efficiency that does not exceed 30%. Why is GUNA flu so extraordinarily effective? Because it does not contain and does not have a vaccination action, but the two NOSODES in particular are able to stimulate these immunocompetent cells, the cytotoxic T lymphocytes and the natural killer cells. These two immune system cells can lyse the cell infected by the virus. Therefore, if the virus changes its conformation, it doesn't matter to the two immunocompetent cells because their action is directed against the cell. By destroying the cell, they remove the sustenance for the replication of the virus, and we might say that they couldn't care less about the drift or shift that a virus can achieve. Let's remember one thing. NOSODES carry out an immune stimulation of a cell-mediated type. The third players in the anti-homotoxic pharmacology represent another extraordinary class of remedies, the so-called SUIS organ preparations. Here we're dealing with pig tissue and organ derivatives. Why the pig and not another animal species? Well, we know that the pig is a species most similar to the human species. This similarity is not only macroscopic. We all know that the cardiac valves of pigs are used, or that the dimensions of the pig liver are very similar to those of the human liver. And this macroscopic similarity doesn't interest us. We are more interested in the immunological type microscopic similarity. In short, the surface antigens of pig organs are very similar to ours. This characteristic determines the so-called organotropism, that is, by introducing an organ preparation into the human body, either parenterally or orally, the homologous or human organ attracts the product, the pharmaceutical, towards itself. This is the first characteristic, organotropism. The second characteristic of pig organ derivatives is intuitive. The proteins of these organs are very poisoned, and we are speaking in fact of adipose degeneration. That is, we can say that they are imbibed with fat. So we are faced with a pharmacologic structure that is on one hand toxic, and on the other hand has the characteristic of organotropism, i.e. to direct its action directly towards a specific target. Therefore, what occurs when using an organ preparation? It happens that with an immune-type mechanism, which we could describe as sub-inflammatory, the organ preparation stimulates the function of the target organ. For example, let's say I have a patient with a hepatic insufficiency. Surely, SUIS, a pig liver organ derivative with a controlled inflammatory type immunological mechanism that is both subliminal and subclinical, will restart the function of the liver organ. The use of organ preparations is typical in all functional and chronic degenerative pathologies. The organ preparations also possess a trophic and therefore anti-degenerative action. It is able, for example, to counteract fibrosis. I would like you to focus on this rule for the use of SUIS organ preparations. Remember, when a SUIS organ preparation is used at low concentrations, for example X6, it has a stimulating effect on organ function. And when it is used at a high homeopathic dilution, for example X200, it has an attenuating action on organ function. A very simple, absolutely textbook example is that if we are faced with hypothyroidism, the use of X6 thyroid gland will be indicated. But if we are faced with hyperthyroidism, the use of X200 thyroid gland will be indicated. Let's remember this rule because it will allow us to understand how in some P.R.M. pharmaceutical compounds, simple dilutions of organ preparations or a particular pharmacologic form that we call according potencies are present, while in the same substance derived from the pig organ or tissue, the low dilution X6, the medium dilution X30, and the high dilution X200 are present together. In this, the action is neither of total stimulus nor total attenuation, but of regulation of function.